Welcome, everybody. We're so glad to have you here for another talk out of the series Live Longer, Live Stronger in partnership with the Sanford Burnham Prebys Medical Discovery Institute. This has been a wonderful partnership for us with the Medical Discovery Institute, and we're glad to be able to offer this 5-part series. And if you were following the PowerPoint presentation at the beginning, we have an exciting event coming up for everybody who has purchased a ticket for this event series. If it was one event or more, doesn't matter. You will be invited to join us on May 26th for a very special forum series that will bring in back all the speakers that we had for the series. And then we put them into small breakout rooms, and you can have individual conversations with the speakers around the topic that you're interested in and that you joined the event for. So hopefully that will be something that you look forward to just as much as we do, because it's always good to have a more personal conversation and get your questions answered on any given topic. I have some good news to share in addition to that. The fleet will be reopening on Friday, April 9th. Yay! It's been a year that we've been closed, so it's just amazing. We're going to be open from Fridays through Mondays, so it's a shortened week for now. But we hope that you'll come visit us with your family again. We can assure you that we're taking all the safety precautions. There will be mask wearing in the house. We'll do extensive cleaning in the building. All of our staff and volunteers will get a health check when they enter the building. So we'll do whatever we can. You can purchase your tickets in advance online, so you don't have to wait in line. We're doing everything we can to keep everybody safe, but still having a good time and enjoying some science in the Fleet Science Center. And I think with that exciting note, I'm going to leave it at that. I could tell you much more about programs coming up, but you saw the slides at the beginning, so I'm going to leave it at that. I'm going to turn it over to our wonderful speaker today, which is Dr. Francesca Marassi. And so Francesca, if you want to take it away, that would be wonderful.
Is my screen okay?
Yep. Actually, now we're seeing your regular presentation and the notes on the right side, so it looks a little bit different than before.
It was working so well just a minute ago.
Exactly. We tested it and it was perfect.
Why is that? Okay.
And if you need to stop sharing and reshare, that's perfectly fine too.
Yes. The joys of Zoom.
Yes, the joys of Zoom. While Francesca is getting that ready, just to let everybody know, Francesca will go through her presentation and then we will have time for Q&A. And as you've seen in the presentation that was running before we got started, there's a Q&A function. There's a little button on the bottom of your screen with 2 speech bubbles that you can click, and there you can type in your question, hit send, and then we receive it. And I will help Francesca moderate the Q&A session, so we will have time to hopefully get to all the questions at the end of Francesca's presentation. And while we do the Q&A, I will launch a little survey in form of a poll. So while you listen to the Q&A, if you could take a second to fill out the poll and let us know what you think about the event, who you are, that would be wonderful. Because as you know, in science, it's all about data.
Is this okay?
I don't see anything yet.
Okay, now it's saying that screen sharing has failed. Oh.
Uh-oh.
Okay, hold on one second.
Sure thing.
Everything was so perfect.
It was so beautiful. I guess it's also science. It works perfect in the test, and then you get into real life.
That's exactly right. Apologies to everybody.
It's quite all right.
Okay, that's okay.
Let me know when you
Yes,
think you started sharing, so I can
trying here.
let you know if we see anything.
That's not what I want to do, actually.
And I hope you all will be joining us on May 5th for our last official presentation of this event, where we're going to look at staying strong as we age and take a closer look at bones.
How is that?
That is perfect. So
okay.
Now we can see everything.
Good.
Yay!
So now I need to get back. Okay, here we go. Apologies for the delay. Okay, thank you everybody for joining and for attending this presentation. My name is Francesca Marassi. I am a professor at the Sanford Burnham Prebys Medical Discovery Institute. I will talk to you today about some research that we're doing in the laboratory that's focused on age-related macular degeneration, hence the title, Seeing is Believing, Preventing Vision Loss as We Age. Seeing is believing also has a double entendre here, and the reason for that will become apparent. Is related to the fact that the research that we do in the laboratory is very much associated with looking at things, with seeing the shapes of things. In this case, the molecules, the components that really go and either keep our cells healthy and functional or are responsible for leading us down the pathway to disease. So as a way of introduction, I'll show you this cartoon that basically illustrates very schematically the way in which we see. So you see, I got my mouse here if I can, a nice picture of the eye here. And can you see my cursor or my pointer here?
Yes.
Okay.
Yes.
Okay. So in this nice cartoon, you see the light coming in and hitting your pupil, your iris, your pupil, your lens. What happens then is that the light basically gets focused into this spot called the macula, where you have basically a concentration of photoreceptor cells, an retinal epithelial layer shown right here in the cutout that is responsible really for detecting lights and for detecting and perceiving the images that you see. So this little box, all of these layers are really composed of this very intricate network of layered cells that include the photoreceptor cells that are responsible for transmitting the light and visual impulses that you see, a retinal epithelial layer. These are these fat yellow cells that you see right here. And these are really responsible for providing this barrier between the eye and the blood vessels. And then a choroid layer of blood vessels that is really responsible for supplying the retina with nutrients going in. This should be reversed, waste going out, and maintaining the proper temperature, volume of the eye, maintaining a healthy eye. Okay. So this choroidal circulation really accounts for about 85% of the total blood flow in the eye. The exchange across this barrier needs to be maintained for vision and everything to be healthy. Okay, so this exchange of nutrients and metabolism really across the barrier is super important. So what happens as we age? As we age, this region of central vision, the macula, begins to accumulate abnormal deposits, okay, also known as drusen from a German word. And so what this does now, if we look at a zoom in view of the picture, of the eye picture, is you end up basically gumming up this permeable layer that is important for nutrient exchange across the eye barrier. And so what happens is that the metabolic exchange is impaired, the layers of the macula, including the photoreceptor cells and the retinal pigment epithelial cells, get progressively thinner, begin to atrophy, and this, of course, compromises function. These deposits themselves may lead to deterioration and atrophy of the retina, and the degeneration is unfortunately accompanied by impaired vision and ultimately blindness. So I have here two pictures that give you an idea of what it might be like to have this terrible disease, which unfortunately afflicts the majority of people over 65, okay? So the mechanism of disease progression is not well understood. As of August 2019, there's about 110 or so clinical trials in various stages of progress. There is therapy available that addresses the wet version of macular degeneration, in which blood vessels, in which the macula is highly infused with blood vessels. But the majority of AMD patients, 90% of AMD patients, have a dry version of AMD. This is the most prevalent form, and this is poorly understood, and there's no therapy available for that. So little is known about the disease progression, but what is known is that these deposits, shown here as these dark spheres that accumulate under the epithelial layer, are not only a hallmark of macular degeneration, but also a hallmark of Alzheimer's disease. And they also share similarities with the plaques that form in atherosclerosis. So all of these are diseases that are typified by the deposition of, protein, biomineral, lipid deposits. And so understanding exactly how these deposits form, what their composition is, and how they form is important for developing therapy, not only for macular degeneration, but also for these other major and devastating diseases, okay? So how might you see these deposits? So they're known to be highly heterogeneous, okay, composed of many different kinds of components. They're rich, for example, in blood proteins, in lipids, and in things like calcium and phosphate, and I'll be talking about that. There are several of the proteins that are associated with the deposits are involved in immunity. And this has been proposed to reflect a kind of immune response to some unknown stress cues that accumulate as we age. The deposits can actually be imaged with a number of clinical and laboratory methods, and these reveal the heterogeneity, and some of the examples are shown in this image, okay? So in this retinal camera picture here, which really involves photographing the rear of the eye, also known as the fundus. The yellow spots identify the drusenoid deposits that are a hallmark of macular degeneration. Okay, so if you go to the ophthalmologist and you get a picture like that, that's not a good sign. Okay, lots of yellow spots, lots of deposits. In optical coherence tomography, what we're doing is actually imaging the epithelial, the layer between the epithelial cells and the choroid, and the deposits really show up as these ruffles in the layering that begin to intrude into the eye. Okay, so you get these pretty pictures of the drusenoid deposit formation. You have different kinds of stains. For example, this hematoxylin or eosin stain, the drusen appear here in these sort of pinkish spherical objects in the same position between the two layers. Autofluorescence, also these spherical globs, and von Costa stain, also these spherical globs. Okay, so the point here is not really to understand what all these stains do, but to show that there's many evidence, there's many types of experiments and diagnostic tools that we can use to look for these kinds of deposits. But the question is, how do they form? I mean, imaging the pathology is obviously critically important for therapy for diagnosing. But for therapy development, we really need to also understand how these deposits, what is the composition of these deposits, and how they actually form. Okay, and so what we mean by that is an understanding at a more molecular scale. Okay, so 1 billionth times smaller than what we're looking at right here, if you can imagine that. So what I mean by that is this, for example, as we begin to go down and zoom in on these smaller scales. So we go back to the picture of the eye. If I take a cutout of the macula, I begin to be able to see the layers of cells, the photoreceptor cells, epithelial cells, the blood vessels, and so on. If I then take an image of these actual deposits, as was done in 2015 by our collaborator, Richard Thompson, we begin to see that the glob of deposits are actually formed by these very pretty uniform spheres that almost look like Lindt chocolate bonbons. Okay, they have all these multiple layers that stain depending on their composition. And to give you an idea of the size of these deposits, now these are about 1,000th of a millimeter compared to an ant, a small ant, for example, that is 2 millimeter. Okay, so now we're beginning to go to a very small scale. So the remarkable discovery here was to show that these globs are made up of such beautiful and uniform structures. And so what they did actually for this experiment is they extracted drusen deposits from AMD patients in the laboratory, and then they milled them. And I actually mean they milled them to reveal that they're composed of these structures. Okay, what are the structures made of? Well, it turns out that the core is lipid or cholesterol, so fat, basically. The pink layer that surrounds it is basically bone, is calcium phosphate, and it's mineralized calcium phosphate. So this is the major component of bone and teeth. We all have soluble calcium. We have soluble phosphate in the body. It doesn't just naturally mineralize, otherwise we would all be dead. It's a highly regulated process typically. But in this case, there's some abnormal process that occurs to generate the formation of bone on the surface of these fatty lipid and cholesterol particles, okay, these lipid droplets or lipoparticles. On the surface of that, there's another coat of protein. And in these three images, the protein is colored depending on the protein type, okay? So it's not important to know what they are right now, but as you see, you have amyloid beta in red, vitronectin in yellow, complement factor H in green. The point being that all of these are proteins that are enriched in blood, and they all are found on the surface, like coating the surface of these spherules. So this really provided a detailed analysis of the constituents of the AMD, of the macular degeneration drusen, and it really gives us a handle on first of all, proposing a mechanism for how they might form, and then down the road, suggesting ways in which could perhaps prevent them if we could just begin to handle the proteins and interfere with the way in which they operate and associate with the lipid and with the bone component. Okay, so in terms of a mechanism, well, based on these fears, one can propose that they might form by first mineralizing some bone, a shell of bones around the fatty cholesterol lipid droplet. And then these might accumulate with age under the epithelial space and eventually cloak themselves with a protein to aggregate, to further aggregate. So you can see, for example, here in the LTI, metabolism is perfectly fine across from the blood vessels to the retinal epithelial layer. But as we age, if metabolism becomes impaired by some unknown stress, then you may get the accumulation of lipid droplets. Okay, so these are really like little globs of fat, of lipid, cholesterol, that may begin to be retained in the space under the epithelial layer and between the blood flow. Eventually, these basically serve as seeds for the formation of bone around them, and then these get cloaked with protein shown in blue, which begin to aggregate around the seed and form the deposit. Okay, so that's a nice model, but without knowing the details, it doesn't tell us much. There's obviously a lot of work to do. And part of the work has to entail looking in detail at the molecular components. Okay, so the protein, the lipid, how do they all actually work together to begin to form these kinds of structures that then aggregate and gum up basically metabolic exchange across this epithelial layer. So in my laboratory, we became particularly interested in this protein vitronectin because amongst all of the images, it was unique in forming both this very even coat that you see, this yellow bright corona around the sphere, but also because it has these very diffuse flower-like petals almost around the sphere. And what they suggest to us is that we've sort of captured vitronectin in action. We may have captured vitronectin as action as it is actually coordinating the formation of the spherule. And so if vitronectin is indeed a protein that coordinates the formation of these spherules in the AMD deposits, then if we could perhaps understand how it does it, we might then have a way of interfering with the activity. So a little bit about vitronectin. It is a multitasking blood protein. It's abundant in blood. We all have it. It circulates. And it has multiple functions. So it's important for lipid storage and transport, so trafficking lipids across different parts of your body, depending on metabolic need. It's very important for maintaining a healthy immune system. It plays a role, unfortunately, in cancer metastasis and cell migration. And it also is important for pathogen, host pathogen interactions. And actually, in one other project in the laboratory, we're trying to understand how vitronectin interacts with bacterial pathogen to facilitate its invasion of the human host. In bold here, I've highlighted the fact that it is a major component of age-related macular degeneration. It's also a major component of the fibrils that are identified in Alzheimer's disease brains. And it's a major component of the deposits that form in atherosclerosis. And so the data really indicate that vitronectin may be more than just a passive component of these deposits, but an actual active partner in the formation. And because it's associated with these other diseases, then understanding exactly how it plays its functions, it's important. It's important because it's interesting, but it's obviously important for therapy development. Okay, if we know what it does, if we know what it looks like, then we can perhaps develop a drug. Okay, so what do I mean by structure? What do I mean by function? Okay, we want to know fundamentally how does it work, and the philosophy of my laboratory is basically to say that if you know the form, if you know the structure of something, then you know the function. Okay, so to know the function, you want to know the structure and vice versa. And so here are some examples from everyday life. How does a clover knot work? How might you go about making such an object? If I asked you that question without showing you the picture, you might just look at me blankly. You would never know that. But if I show you the picture, well, you can get an idea of how you might go about and make it. Same if I ask you, how does a propeller work? Well, what do you mean by that? Do you mean a helicopter? Do you mean a propeller in a helicopter, a propeller in a plane? It's very different. So if I show you a picture, then you might begin to tell me, well, one propels forward and one propels upwards. So seeing things is important. And that's really what we strive to do in the laboratory, except that instead of doing it with tools and objects and arts and crafts, we do it with genes and with molecules, okay? So in the case of the knot, we go about asking, well, how do we go about taking a string that's maybe colored in 4 different parts and folding that into a knot? The analogy in the laboratory is to take a gene that may be perhaps made up of different types of different genetic elements, of different sequences. How do we go from a linear gene like this into a protein that's folded? And so Kyung Shin, Mia Fujimoto, and Bernard Lechtenberg did exactly this in the laboratory. They started out with a gene that encodes all of the information for the sequence of vitronectin. So the gene carries the genetic information for actually making the protein, which is made up of amino acids. And so they went from the gene to the protein. They were able to fold it just like a knot, and to show that the structure actually looks like a 4-bladed propeller. Okay, it's very pretty. It's a flower-like structure. And I'll show you a little bit more detail in this movie. Okay, so if you take the structure, the shape of human vitronectin, this is actually what circulates in our blood. You can see that it's made up of these 4 different leaves, and I color them by leaf: blue, green, yellow, and red. They are circularized, basically. So they click closed at the end here. You can see this yellow stick that's basically a little latch that closes this flower-like structure. So what's interesting about it is, of course, it's very pretty, but also there's a channel in the middle, and the channel is filled with salt. It's filled with 2 atoms, 2 spheres of sodium, and 1 cyan sphere of chloride. Okay, so they fit perfectly inside this channel, and they provide structure to the protein. And they also end up being carried throughout the blood by the protein. So in this different representation now of the protein, what we're looking at is the surface that other blood components might actually encounter when it's in blood. Okay, so the surface representation now is important because it gives you a feel of the various nooks and crannies that the protein presents, and in which parts of it might actually act as little binding pockets for other partners. So it's not a smooth sphere, right? Like we're not encountering just this smooth marble that is not sticky, but you see right here, for example, this beautiful groove that's almost like waiting for a partner to come in and bind to it. There's another one on the other surface that's colored especially white, meaning that it's very sticky. Okay, so red means it's negative charge, blue means it's positive charge, but white means that it's ready to bind things like lipids and very sticky proteins. And indeed, vitronectin is one of these proteins that's also a major component of HDL, okay, high density lipoparticles. So to further understand now what the function of the protein might be in the context of macular degeneration, we turn to a technique called magnetic resonance. I think you all know this because many of you will probably have had an MRI, okay, a magnetic resonance image. So in MRI, typically your body gets inserted inside the bore of this magnet, okay? So this beer tank or donut looking thing right here, okay? So by doing this, you can image various parts of your body. And what I'm showing you here is an image actually of the eye. So this turns out to be some image of a poor patient who had a contact lens stuck up at the top of the eye, and this could only be detected by MRI eventually. Okay, so we're seeing things in the centimeter to millimeter scale. Obviously, this is a head, so what is that? Maybe 10, 12 centimeters across, something like that, okay? So an analogous technique that we use in the laboratory is NMR, nuclear magnetic resonance. Now, nuclear does not mean that it's radioactive. It's all stable isotopes. But what it means is that we're now looking at atoms. We're no longer looking at bodies, cells, tissues, but we're looking at the sub particles that make up all of the cells and tissues and body parts and organs, okay? And so with this technology here, this happens to be a magnet that we use at UCSD, but we have similar magnets here at the Institute, and we do have open houses, so I would encourage you to come and take a look at the lab if you have an opportunity. So in this case, the magnet you can see is now standing upright. And instead of sliding a body into the bore, we insert a tiny sample of something that we made in the laboratory. It could be a protein, it could be a piece of DNA, it could even be some material that we want to look at the structure of. But the outcome is that now we're looking at sub-nanometer, 1 billionth time smaller components of biomaterials compared to MRI. Okay, so we end up looking at structures of proteins, and here's a nice collection of some of the proteins that we looked at in the lab. It's not important to know what they are. They give very pretty pictures. And the structure is, of course, important because then we could try to understand functionality. So using this nuclear magnetic resonance technology, or let's just call it magnetic resonance for the sake of analogy to MRI, we were able to find that vitronectin is not only a salt binding protein, okay? So sodium, chloride, sodium, there happens to be a water molecule stuck here shown in red and a sulfate molecule stuck here at the bottom, but it also is a calcium binding protein, okay? So calcium can actually replace this salt atom right at the top here. And so this is important because if you recall, I showed you in the beginning that these spherules that make up the deposits of, that are found in macular degeneration are rich in calcium phosphate, they're rich in bone. So the discovery that vitronectin is a very specific calcium binding protein tells us that it, in fact, may be important for orchestrating, for organizing the mineralization of this bone on the surface of lipids. And so the way that it binds calcium is by basically exposing these 4 arms that are showing you in yellow. Okay, so these are specific amino acids that expose out, they jet out on the surface of the protein like 4 arms that literally grab the calcium atom out of solution, stabilize it, and then use it to both associate with the bone and produce it basically and mineralize it. Okay. So this is, by the way, typical of bone binding proteins, having these, these kinds of structures with 4 acidic amino acids that can bind calcium is a very common way for both coordinating calcium and associating with bone and functioning also in things like bone remodeling. So this kind of picture now led us to this model, which we're proposing for moving forward with therapy development. Okay, so what could vitronectin be doing in AMD, in orchestrating AMD deposit formation? Okay, so in one scenario, calcium here shown in green and phosphate shown in red and orange. So in one scenario, vitronectin, by binding calcium, could be seeding these clusters, these bone-like clusters around a lipid droplet shown right here. Okay, and so it could be actually the first step that's required for producing the layer of bone on the surface of lipid. Of note also, most lipids are phospholipids, meaning that they have a phosphate group exposed on their surface. And so the association of calcium that's ligated by vitronectin with these phosphate groups could also be important for initiating the formation of this mineralized shell in the drusen AMD deposits. And finally, I mean, this is perhaps more, a little bit more far-fetched, but potentially could be controlling mineralization and also the stability, let's say, of this shell of bone on the surface of the spheres once they are formed. Okay. But the important thing is that we now have a handle for moving forward with therapy development. So the goal right now is then to, first of all, ask, is there a compound that we can either identify, screen, or develop that will break up this interaction right here, okay, of vitronectin with the hydroxyapatite and the calcium. And one of the things that we are moving forward to do in the laboratory is to see whether antibodies can interfere with this association. So can we identify an antibody, for example, that will bind to this region of vitronectin and interfere its association with hydroxyapatite in bone, mineralized calcium phosphate? And is that antibody then a viable first-in-class compound that one could further develop to begin to interfere with AMD-like deposits? And so in the laboratory, Kyung Shin and James Kent have begun to do these kinds of experiments where they focused on actually making these types of spheres in the laboratory in vitro. Okay, so this is the actual macular degeneration sphere that I showed you before with this beautiful lipid core, bone shell, and protein shell around it. And what they have been doing in the laboratory is generating lipid particles and then coating them with bone from first principles, okay, from calcium and phosphate solutions. And you can see now that they've been able to generate this pretty shell of bone around the sphere, which they're now preparing to test together with vitronectin to see if vitronectin can accelerate this or somehow affect it in a way, and whether in the long run, whether antibodies or other compounds can interfere with this approach, okay? So I think with this, I'm going to leave you or leave the topic. And hopefully, yeah, I hope to have left you with a flavor of the kind of research that we do in the laboratory, of the importance of looking at shapes of things and structures of proteins in this case, and the potential for actual precise therapy development opportunities that this kind of research brings. I'll close off by showing pictures of some of the people who worked on this project, and I already mentioned some of them: Kyung, James, and Mia. Alex Alessian was also critical for a recent structure that I didn't show, in which we showed that vitronectin is bound to calcium, and all of the other people in the laboratory. And thank you very much for listening, and I'm open to questions.
Wonderful. Thank you so much, Francesca, for that presentation. I'm going to stop sharing your screen. There we go. This way we can see you better. And we do have some questions, so I'm just going to read them to you if you don't mind. Mm-hmm. Somebody is stating that clinical studies have shown that having adequate macular pigment in the human retina decreases the risk of developing AMD. Do your findings about AMD explain the protective role played by macular pigments?
They do not, actually. So our research has only been focused on these deposits that we observe, and the types of deposits also that form in the other types of ectopic deposit diseases. So unfortunately, no, I don't have any insights in that regard. I'm hoping that as we move forward, colleagues who work on cell-based models of macular degeneration will be able to address this important question.
Thank you. And for everybody who just launched the poll, so if you can take a second while we go through the Q&A to take the little survey, I really appreciate it. And there's another question: the autofluorescence image of the AMD, is that live or fixed tissue?
This is fixed tissue.
And can you explain a little bit more about that?
Let's see. Let me see if I can go back to it. I think you're talking about this one. So the autofluorescence, I think you're talking about the autofluorescence image that's being shown right here.
Yes.
So it's showing basically this more or less circular drusen or deposit that's being formed. And so the autofluorescence of the retinal pigment epithelial is more intense and yellowish, whereas the autofluorescence here that's greenish is what's called the Brooks membrane, which is really this extracellular matrix layer that separates the epithelial cells from the choroid, from the blood vessels.
Thank you. Appreciate that. We couldn't see the image, but I think it's still.
Okay. Can you see? Oh, because you're not seeing. Oh, right.
Exactly. I'm not sharing it more. And Rebecca is wondering, where do you get samples from to initially see the layers in the eye?
Well, unfortunately, the spherules that I showed you that came from our collaborator study, Richard Thompson, these were actually taken from cadaver eyes. And so they collected these deposits, and then, as I said, they milled them. But what I actually mean by that is that these are very tiny things, so you cannot go in and mill them with a drill. You go in and mill them with an ion type drill. Okay, so you focus a very, very strong ion beam at them, and basically evaporate off the surface layers until you reveal that spherical structure that I showed you in the pictures.
Wow. That sounds fascinating. And she was also wondering, where do you get cells to do your lab tests for NMR and growth? And what is NMR? I have to ask.
Okay. So what is NMR? Okay, so the best analogy is to MRI. So it's a big magnet, okay,
Oh, it's the shortened version for the machine that you showed us.
Yes, that's right. It's
yeah.
two types of technology. Okay, so it's all magnetic resonance. So in one type, MRI, you do imaging. In NMR, you kind of do imaging, but you pictures of much smaller molecules. And so in my laboratory, rather than looking at cells, we actually look at proteins, and we look at assemblies of proteins, not so much as cells. Okay, so I was trying to convey the idea that with the two techniques and these other autofluorescence and staining techniques that I showed, we had the opportunity of going from large scale objects like cells to very small scale objects like actually the components of cells, so the proteins and the lipids and the bone particles that come together to make the various structures that we have in our body. And so in my laboratory, we work in vitro. And what that means is that we make the proteins that go into cell, we produce them, and we put them in test tubes so that we can use NMR spectroscopy, NMR techniques to look at the structures. The way NMR works, so you're basically placing your samples inside this big magnet, and then you apply radio waves to stimulate different parts of the sample to give you signals that you can then correlate back to the structure. That's kind of a quick explanation.
So you apply some radio waves in some part of
it's the same as MRI.
And you see the wiggle.
kind of like that. Okay, so it's sort of the same as an MRI. So when you're sitting inside an MRI machine, if you've ever had an MRI, you hear this. They have to give you. You have to cover your ears, right? So you have these incredible sounds that come from the impulses that the machine sends out. So we do the same, except we don't do it to bodies, but we do it to little samples of proteins.
I appreciate that. So I don't know if this works. I have two folks who have raised their hand, and normally, with the webinar function, we don't have audio or video for our participants, but I'm going to try right now if it works. So Deborah, I'm not sure if you can speak now. If you want to try and ask your question. I assume you have a question. You raised your hand.
Can you hear me?
Yes.
Okay, so my question is, what can someone do to prevent macular degeneration? Like, are there a healthy diet or exercises, or,
Yes.
I really didn't understand. Actually, this kind of just all went way over my head. Like, what can I do to prevent macular degeneration?
Well, okay, so there are risk factors. Unfortunately, age over 60 is a risk factor, but that's not a death knell. History of smoking is a risk factor. Family history and genetic predisposition is a risk factor. And so the things that you said, healthy diet, exercise, not smoking, living well, of course it is. And especially so with some of the things that we talked about, the fact that it's clearly, there clearly is an association with metabolism, right? Like lipid trafficking. So healthy diet, healthy exercise definitely have an impact on macular degeneration and obviously all of these other major diseases. Having said that, it is a devastating disease, and unfortunately, there is no therapy especially for this dry version that's highly prevalent. And so I'm hopeful that as we learn more about the various components, that it will be possible to develop therapeutic compounds, drugs, that will be able to relieve the disease.
So right now there is no treatment available, or no compound that would even prevent getting the disease.
Not prevent, no. So there are, you can inject, there are some therapies, they're injectable, so they're eye injectable therapies. that are targeted for this highly vascularized version of the wet type of macular degeneration, which is unfortunately it's less prevalent than the dry version.
Thank you for that. Is AMD, can I pass it on to my children? Is it inheritable?
So the genetic component is not clear. It's not clear what the gene is. There are recent studies showing panels of proteins and genes that appear to be highly associated with it. So there are some polymorphisms, some genetic mutations that are inheritable and that are associated with AMD. It appears to be, but there's little known about that just yet. The research is still very young.
I'm going to keep up with that. My mom has the problem, so I'm hoping that I don't get it either. I'm going to try this one more time with the raise the hand function because Gary has the hand raised as well. This is new to me, so I apologize if it's not going well. Gary, can you maybe try to talk?
Yeah, the lady before me asked my question. It was, what can I do? And that's pretty much answered.
Oh, perfect. I'm great. Thank you.
I wish I had the key answers for everybody. I think for now, yes, healthy diet, healthy lifestyle.
Yeah,
is the key.
Okay, thank you.
Thanks, Gary. And then I was wondering, you had mentioned in your talk that the lipid that is causing this is also responsible for Alzheimer's and others. So if you find a compound that works, do you think the compound might also work for other diseases that this lipid is responsible for?
So that's a stretch. I mean, that would be overselling it for me to say yes. But I think that yes, I mean, what I'm hoping that I did is also to give you maybe a flavor of how some of the science works, okay? And that is by knowing how these components get together and work together, then we begin to get insights about the same things that they might do in different kinds of diseases. So for example, vitronectin forms fibrils. There's actual pictures of Alzheimer's brains with vitronectin fibrils. So these, entangled strings of vitronectin, okay, that somehow unfolds and forms in them. So I think by learning more and more what it does and potentially developing compounds that work for AMD, then yeah, who knows? Maybe that we will have a handle for those kinds of diseases as well. We will certainly learn something that will be valuable for those pathologies in addition to AMD.
Yeah. We have somebody here who says she was told that she has the wet kind and she tears up often. She takes medication called preservision. And do you have any insight on if that is helpful?
So there are, I don't know about this particular medication. So there is some so-called anti-VEGF therapy for wet AMD, and I'm not sure if you're getting the injections or not, if these are just eye drops. For more severe forms, there are some injectable antibody-based compounds that can be beneficial.
And those would be in an eye clinic, somebody would inject those?
Yes.
Yeah.
Yes.
When you talk about compounds, what do you think they might look like? Would it be something that's more in a pill form, or is it something like an injection that would have to happen?
Yeah, so most therapy for the eye, so the eye, like the brain, is an immune privileged area, okay? So meaning that they are sort of walled off by barriers, by these membranes, like I showed you, this Brooks membrane and the epithelial layer and so on, that sort of isolate it from the rest of the body, okay? And so what it means is that getting drugs across those barriers is not necessarily straightforward, okay? So in the case of AMD, for example, like I said, the anti-VEGF therapy for wet type AMD is an injectable. Okay, so it is something that you do in the clinic. And in the case of dry AMD, I mean, obviously we're not anywhere close to developing the therapy, but the goal right now is to see whether we can come out, for example, with an antibody that will block the association with these deposits. And if that's the case, then yes, I envision that it would be, the formula would be an injectable type of therapy. Okay, so the compound would be the antibody in this case.
And can we imagine that like an antibody that we would get injected with, say, for the COVID vaccine, you know, or treatment, or is it different than that?
It's different, okay? So that would be different. Yes, it would be a different antibody, but you're right. I mean, the idea is the same, okay? The idea is the same. Even, you know, antibodies that are raised in response to COVID vaccines, same idea, right? You have an antibody that goes and binds to the offending protein and it stops its function. You're basically like, you know, splatting a bunch of glob on top of the protein so that's it. It cannot work anymore. So that's the idea, okay? And of course, I mean, that's pretty crude, but you want to try and identify those active sites so that you basically cover them up and that's it. They cannot function anymore, or they cannot be detected anymore by other parts. Yeah.
I think that's a great way to explain it. I like that.
Okay.
And we've got some comments because we had talked earlier on a question on macular pigment and how people who have low levels of the pigments are at possibly a higher risk for developing AMD. And it says here that humans cannot synthesize these modules, but must ingest the plants that contain pigment precursor molecules like lutein and I'm not going to even try to pronounce that word. And apparently the preservision is a supplement that contains those macular pigments. Do you have any comments on that, Francesca?
I don't actually. This is something I didn't know. So it's very interesting. I think it's again, it speaks to the wet version of AMD, not to the dry, and to the formation of these deposits. But yeah.
So more research has to happen to really answer those questions, right?
Yes, that's definitely true. That's why we're here.
Exactly, that's why we're here. And that's why I'm just super thankful to have such institutions in San Diego that do such amazing research, like the Sanford Burnham Previs Medical Discovery Institute. And of course, there's others also, but you all do amazing work on so many diseases and issues that society is facing. It's very fascinating to hear.
Thank you. Thank you very much.
Francesca, thank you so much for your talk. We're just at time, so we're just at 3 o'clock, and I want to keep to everybody's time. I know days are filled up with lots of activities and schedules. I thank everybody for joining us today. Thank you, Dr. Francesca, for a great talk.
Thank you, Andrea, and thank you everybody for the questions.
And thanks everybody for joining us today. I do hope you're going to join us next month again for the Live Stronger series that we have. And we're actually going to look at skeletal muscles. I made a mistake when I said we're talking about bones, and I apologize for that. It's all correct on the website, but of course I had to mess it up today. So we're going to look at skeletal muscles when we do our final talk. And please keep your eyes out open for the email that's going to invite you to that exclusive forum event. We hope you will join us and have a conversation with the scientists. And please do come visit us at the Fleet Science Center. We're so excited to be open again on Friday. So Friday, 10 a.m. Get the family together and come visit us. We promise we make it fun and safe and enjoyable for everybody who would like to join us. And with that, I thank you and close out this talk. We're going to post the video of this talk at the Fleet Science Center YouTube channel at one point. It's probably going to take about 2 weeks or so, a week or two, but you will be able to share this video if you enjoyed it with your friends and family. With that, I wish you a wonderful rest of the week and stay safe and stay healthy.
Thank you.
Bye.
Bye.